Good afternoon. So today we are starting the metabolism. We have completed your first year internals portion. We are starting with the second internal portion. So we are coming into the active part of biochemistry, that is metabolisms. Before starting into metabolisms, everything we have to relate metabolism to whatever happens in human body. So before starting let's know what is the definition of metabolism. Metabolism. It is a complex of intricately regulated systems of energy producing and energy utilizing reactions. So metabolism just is not limited to either production or for utilization. It is a combination of both. So there are two phases in metabolism. One is catabolism and another one is anabolism. Catabolism. Catabolism means catastrophe, that is destruction. So it is a process of digestion or utilization of the proximate substances or proximate principles of life, such as carbohydrate, lipids and proteins. Like it is a catabolism of our energy production. The energy is obtained from carbohydrates, lipids and proteins. Next, anabolism. Anabolism means synthesis. Synthesis of new compounds. Synthesis of macromolecules. Because we are utilizing carbohydrates, at the same time we need glucose for energy production. So, synthesis of glucose. Production of glucose. Then, contraction of muscle. Active ion transport. Thermogenesis. All these are anabolical processes. Now, what does catabolism lead to? Catabolism leads to the formation of ATP, NADH and NADPH. ATP is the energy currency of the cell. NADP, from NADH you will be getting energy. NADPH is just used for oxidoreductase reactions. It is just used for oxidation and reduction reactions, and it does not per se, it does not produce any energy. Only NADH, when it enters into the electron transport chain, will be producing energy. What will anabolism do? Anabolism will utilize the energy. So, catabolism will provide the energy, whereas anabolism will utilize the energy, because to synthesize something, you need energy. For muscle contraction, energy is needed. For active transport, energy is needed. So, at the end of anabolism, what are we left with? We are left with ADP, NAD plus and NADP plus. So, this defines catabolism and anabolism. Now, as I mentioned, there are three proximate principles of life. They are carbohydrates, lipids and proteins. Carbohydrates. For human beings, the important carbohydrate is glycogen. The important nutrient is triglycerides. So, because the triglycerides will have glycerol, along with that they have fatty acids also. Then proteins. So, what will happen to the glycogen? The glycogen will undergo glycogenolysis and it will be forming glucose. This glucose will undergo glycolysis and it will form pyruvate. What will happen to triglycerides? The triglycerides will undergo lysis, lipolysis. As a result of that, the glycerol part is removed and fatty acids are available. Now, these free fatty acids will undergo oxidation. So that is the next level. Then proteins. Proteins will undergo proteolysis and form amino acids. So these are the major macro components which are subdivided. These have gone down to few steps to form these metabolites like glucose, free fatty acids and amino acids. Now, from free fatty acids, these free fatty acids will undergo beta oxidation to form acetyl-CoA. Amino acids will undergo transamination, deamination, oxidative deamination. All these things they undergo and finally they form few of the amino acids. If they are ketogenic amino acids, they will be forming acetyl-CoA. Now, the glucose will form pyruvate at the end of glycolysis, that in the presence of PDH complex will be forming acetyl-CoA. So, what is the gist of whole of this slide is that the major macromolecules such as glycogen, triglycerides, protein, all these are catabolized to finally form a main component called as acetyl-CoA. This acetyl-CoA is the one that enters into the citric acid cycle and that will be producing the ATPs. So, whatever components we take, finally they will converge and they will be forming acetyl-CoA so that they can enter into the citric acid cycle. Next, so digestion. Now, in the metabolisms when we are talking, metabolisms we divide into three phases: primary metabolism, secondary metabolism, and tertiary metabolism. Primary metabolism is what happens to the food that we take. So the food that we take is digested. So that is a primary metabolism. Secondary are the intermediary metabolism, is what we have seen previously. What happens after digestion? You get some of the substances. What happens to those substances in the human body? And finally, they form into carbon dioxide and reducing equivalent. So what is included in secondary metabolism? The previous slide that we have seen. From glycogen to acetyl-CoA, that acetyl-CoA entering into the citric acid cycle. From triglycerides to fatty acids to acetyl-CoA, it entering into the citric acid cycle. Proteins to amino acids to acetyl-CoA, and which enter into the citric acid cycle. So that whole component till the end of the citric acid cycle is involved in secondary metabolism. Now we are going to study the secondary metabolism. Then finally is the tertiary metabolism, that is the oxidative phosphorylation or electron transport chain, that is the tertiary metabolism. So we have primary, secondary and tertiary metabolism. Tertiary metabolism we have already done. Primary metabolism independently, when we are talking about the carbohydrate, we learn the digestion of carbohydrate, with lipids, digestion of lipids, and with proteins, digestion of proteins. Now, to start with, so we are starting with the primary metabolism, that is digestion. Now this is a metabolism of carbohydrate. So we'll be looking for the digestion of carbohydrates. Glucose. Glucose is universal fuel of human body. Any cell, if it has to work, it has to have glucose, because it is a, that is why we call it as universal fuel. Next, dietary carbohydrates. The dietary carbohydrates, the carbohydrates that we take in the diet are usually, they can be either polysaccharides, they can be disaccharides, they can be monosaccharides, like in different categories we take. What are the common polysaccharides that we take in our diet? They are starch, glycogen and cellulose. Disaccharides, usually we take sucrose and lactose. These are the disaccharides that we usually consume in diet. And in monosaccharides, glucose, to little extent we don't consume glucose as such. We consume glucose as such in a very little extent. Then fructose, because fructose is present in fruits. Now, digestion, because we put everything in the mouth. So, we have to, and everything digestion, everything stops at duodenum level. At the duodenum level, digestion stops. So, when we are talking about the digestion, we have to extend from mouth to the duodenum. What is the path of the food? Digestion in the mouth. The digestion in the mouth occurs for starch and glycogen. This is done by a special enzyme called as salivary amylase. It is done by salivary amylase. This is an endoglycosidase. Glycosidase word means that any enzyme that cleaves the bonds between two glucose molecules, you call it as glycosidase. So, alpha 1,4-glycosidase. So, the glucose that is present in alpha form, and the bond between first C1 and C4 is broken. C1 of one component with C4 of another is broken down by alpha 1,4 endoglycosidase. We call it as endoglycoside. We call it as endoglycoside because this will be occurring in between, anywhere in between the starch or glycogen molecule. So, the digestion of the carbohydrate starts in mouth itself. The enzyme is salivary alpha amylase. You call it as alpha amylase because it acts on alpha 1,4-glycosidic linkages. It is active at neutral pH, only neutral pH is enough, and it requires chloride ions for its activation. So what will it do? It will randomly break the starch and glycogen molecules. Starch and glycogen are homopolysaccharides. They contain only glucose. So it randomly breaks the glucose molecule. So what will happen? You will get tiny pieces of glucose, tiny pieces of the polymers of glucose. So they can be either maltose, they can be maltodextrose. Maltose means 2 glucose molecules combined together. Maltotriose, 3 molecules of glucose, and dextrins, up to 6 to 7 molecules of glucose bonded by glycosidic linkages. So when the starch and glycogen are broken down very randomly by alpha-1,4 endoglycosidase, it results in the formation of maltose, maltotriose and dextrins. Now the next step is its entry into the now, the important thing is that this cannot act on alpha-1,6 linkages. It cannot break alpha-1,6 linkages. So, wherever branch points are there, those are not broken. Next, digestion in the stomach. Digestion does not occur in the stomach because neutral pH is required for all the neutral pH is required for the enzymes that have to act on carbohydrates. Digestion in intestine. The digestion in intestine is mainly mediated by pancreas. We have pancreatic alpha amylase. Same as that of the salivary amylase, pancreatic amylase and salivary amylase, both of them are isomers. So, pancreatic alpha amylase. This pancreatic alpha amylase is the one that helps in digestion and it further breaks down those dextrins, maltodextrins into further smaller particles. Its function and everything is exactly same as that of alpha amylase of salivary gland. Very important are intestinal brush borders producing enzymes. They produce maltase, isomaltase, lactase and dextrinase enzymes. So these enzymes are the ones that will finally break the bigger and complex carbohydrate molecule into very tiny glucose molecules. So digestion, any type of digestion does not occur in the stomach. In the intestine, in the presence of pancreatic alpha amylase, these are further broken down. The final act is done by intestinal brush borders which produce maltose, isomaltose, lactase and dextrinase enzymes. So these intestinal brush border enzymes, they are located in the brush borders. They are protected from getting degraded by means of the glycocalyx. So, maltose. Maltose is acted upon by the enzyme maltase. It produces two molecules of glucose. Sucrose. It is acted upon by the sucrose to form glucose and a fructose. Lactose. It is acted upon by lactase to form glucose and galactose. And all of these are finally converted to glucose. Now, these fructose and galactose are absorbed at the GIT, but in the liver they are converted to glucose. So, finally, a part of it is broken down randomly by the salivary amylase. Then, further it is broken down by alpha amylase of the pancreas. Then, finally, into glucose is done by intestinal brush border enzymes. So, maltose, sucrose, lactose, they are further degraded by maltase, sucrase, lactase to form corresponding monosaccharides, and the fructose and galactose that are formed, they are also converted back to glucose in the liver. So that means that now we are till this part. So it is absorbed as glucose, it is absorbed as glucose and fructose, this is absorbed as glucose and galactose. After entering into the circulation, this conversion to glucose takes place. Now, isomaltose. So, I have named other terms also like isomaltose. Isomaltase is broken down by isomaltase to glucose and to two molecules of glucose. Alpha limit dextrins, small dextrin molecules, they are broken down to maltose, maltotriose and glucose like that. Now, once again, this maltose is acted upon by the maltase to form two molecules of glucose. Then, cellulose cannot be digested by the human body due to the absence of beta-1,4 glycosidic linkages. Why we cannot digest it? Why humans cannot digest cellulose? We cannot digest cellulose because we do not have an enzyme that is able to break beta-1,4 glycosidic linkages. Now, cellulose has beta-1,4 glycosidic linkages. Those cannot be broken. That is why we cannot digest cellulose. But the cellulose that is present in the diet acts as roughage in the body. The fiber that they tell you have to eat more of fiber and all that, that fiber is nothing but the undigested cellulose. Now, so this is about the digestion. Digestion has taken place. Now, the glucose is present in the lumen of the intestine. So, that glucose has to absorb. Finally, the thing you have to remember is that though we consume in the diet, we consume polysaccharides. We consume polysaccharides, we consume a little bit of disaccharides like sucrose and lactose, but most of the times we consume only polysaccharides. But after they undergo digestion and absorption into the intestinal epithelial cell, only monosaccharides are absorbed. So these polysaccharides are broken down, they are digested to form monosaccharides, and only monosaccharides are absorbed. So what are the monosaccharides that are absorbed by the GAT are glucose, fructose and galactose. From mucosal epithelial cell to portal venous system, that is what we call it as absorption. Galactose, glucose and fructose are the only monosaccharides that are absorbed. Whatever complex carbohydrate you take, these are the monosaccharides that could be absorbed. There are two mechanisms by which this absorption takes place. One is active transport, that is which occurs against the concentration gradient. The other one is facilitated transport. This facilitated transport, we'll see what is this facilitated transport. Now, the first active transport what I have mentioned, this active transport helps in entry of the glucose that is present in the lumen of the intestine into the epithelial cells. Don't look into the complexity of this picture, just follow the order. So this we are talking, this is the intestinal lumen. After digestion, we have glucose molecules that are present in the intestinal lumen. Now these have to pass through the epithelial cell so that they can enter into the bloodstream. For this purpose to happen, you have a special receptors called as sodium dependent glucose transporters. These are called as SGLT, sodium dependent glucose transporters, in which entry of one molecule of glucose is associated with entry of two molecules of sodium. Entry of one molecule is associated with entry of two molecules of sodium. So it is a symport of sodium and glucose. But once the sodium enters into the epithelial cell have to maintain its electrogradient. So in order to maintain the electrogradient, what are the sodiums that have entered? They are being expelled out. And we know sodium potassium pump, where 3 sodiums are expelled out, 2 potassium will enter in the presence of sodium potassium ATPase enzyme. So that is why we call it, that is why we consider this as an energy process. We consider it as an active transport because energy is being utilized. So there is a transporter, that is SGLT transporters, that will transport glucose and 2 molecules of sodium. They enter into the circulate, they enter into the epithelial cell. Now the sodium that is present is exchanged, 3 sodium molecules are exchanged with 1 potassium, and which requires energy. Now the glucose that is present in the epithelial cell will go into the portal circulation in the presence of a transporter that is named as GLUT2 transporter, which is called as GLUT2 transporter. Now, glucose transport from the lumen to the capillaries. This is what we have seen. Sodium and glucose, they enter into the circulation. Then after that, the sodium has to be expelled out in order to maintain the electron neutrality of the cell. So 3 sodiums will go out and 2 potassium will enter into the cell. So this will maintain the electrochemical gradient of the cell. Now, the glucose that had entered into the intestinal cell, it has to go into the bloodstream. That is done via the GLUT2 transporters. In the intestine, you have special transporters called as GLUT2 transporters. These GLUT2 transporters will allow the glucose entry into the circulation. Next is passive transport or facilitative diffusion of the glucose. So once it enters into the circulation, from the circulation the glucose is distributed to all the cells of the body by means of passive transport, because glucose is having higher amounts of glucose is present in the circulation. So little bit of glucose can enter along the concentration gradient into the cell. So these are called as glucose transporters. The mechanism is called as ping pong mechanism. So what will happen? So this is a receptor. If you see the GLUT receptor, what will happen? It is open at one end and it is closed at one end. Now, when it is called as T1 phase. Now, when glucose enters and binds to that site, it will bring about conformational changes. As a result of that, glucose is pushed into the cell, and once again it goes back to its T1 position. So, the receptor will be glucose transporter, that is GLUT receptor will be in a normal fashion. Once it binds to the glucose, it undergoes conformational, the glucose binding of the glucose results in conformational changes in the receptor, and this channel receptor or this transporter will push glucose inside the cell. It gets converted from T1 to T2 form and pushes the glucose into the cell. Now the glucose has entered into the cell, and once again the receptor has got its the diffuser, once again the transporter has got its previous shape. So in that way this will continue. This is called as ping pong mechanism. So we have few number of glucose transporters that we shortly called as GLUT1 transporter. The GLUT transporters, we have up to 7 types of GLUT transporters. The GLUT1 transporter, this is mainly present in RBC, brain, kidney, placenta and retina. GLUT2 transporter, it is present in serosal surface of intestinal epithelium. We have seen on liver and beta cells of pancreas. They have got very less affinity for glucose. Next, GLUT3 are T3 transporters. The GLUT3 transporters are present in neurons and brain, and they have very high affinity for glucose. Even though the glucose levels are very low in the blood, they try to pick up glucose in that situation also. GLUT4, they have included GLUT4, then after that GLUT4 is being removed. Then GLUT5 is nothing but the transporter in which we have seen that sodium glucose transporter was for glucose to entry from the lumen into the intestine. In the same way, GLUT5 is sodium independent fructose transporter, where without sodium dependence the fructose will enter into the intestinal epithelium. And the same transporters are present in testes, sperms and kidneys also. Next is GLUT7. We don't have GLUT6, GLUT7. Now we have GLUT4. I will tell you the details of GLUT4. We don't have GLUT6. The GLUT6 is being removed. Next is GLUT7. The GLUT7 is present in liver endoplasmic reticulum. So in the liver endoplasmic reticulum, the cell has got endoplasmic reticulum. From the endoplasmic reticulum, for the glucose to enter into the cytosol, you require GLUT7 receptor. So this receptor is present inside the cell organelle. It is not present on the cell, but it is present on the cell organelle. So these are very important to remember. GLUT, that is glucose transporters. GLUT1 is present in RBC, brain, kidneys, placenta and retina. GLUT2 is present in serosal surface of intestinal cells. It has got very low affinity for glucose. GLUT3 is present in neurons and brain. GLUT4 is present in adipose tissue. It is present mainly in the adipose tissue. Next, GLUT5 is present in small adipose tissue and in the muscle. GLUT5 is present in small intestine that will help in transport of fructose from the lumen into the epithelial cell. GLUT7 is present on endoplasmic reticulum. From the endoplasmic reticulum to the cytosol, the glucose is transported. Now we talk about the GLUT4 receptors. GLUT4 receptors, these are present in skeletal muscle and heart. What is the importance in the adipose tissue? Most important is adipose tissue and skeletal muscle. What is the importance of this? Normally, the receptors are internalized in the cytosol. The receptors are not present on the surface, they are internalized on the cytosol. Once insulin sits on the receptors, this will mobilize the receptors, and these will come out and these will merge with the cell wall. As a result of that, glucose will enter into the cell. So that is what is GLUT4 receptor does. Normally the GLUT4 receptors are not present on the surface of the cell. They are internalized. These are not present on the surface of the cell. These are internalized. So when they are in presence of insulin, this will get activated. They will get opened up and they merge with the cell wall. Now in the cell wall, the glucose receptors are there. Now from that, the glucose will enter into the cell. So here what we have to understand is, for these receptors to get open up, insulin is very much required. That will be the problem. That would be the problem with the diabetics, because they have less amount of insulin. As a result of that, so much of glucose cannot enter into the heart and in the muscle, into the adipose tissue and also in the muscle. Now, the disorders that are associated with the carbohydrate metabolism. The disorders that are associated normally when carbohydrates are not digested because of deficiency of the enzyme, or because of less amount of enzyme, or because of any reason, if carbohydrates are not digested, it will result in bacterial fermentation, especially in the large intestine, and produce gases like hydrogen and carbon dioxide. Along with that, it also produces acetic acid, butyric acid and propionic acid. These acids are osmotically active. So what will they do? They will draw more and more water with it. Because of this, it will result in abdominal cramps, flatulence, diarrhea and dehydration. These are the things that will cause, because if the carbohydrates are not digested properly, because this being carbohydrates very good medium for the bacteria to grow, the bacteria will further ferment the glucose to form hydrogen and carbon dioxide. So these are the disorders that we see with the carbohydrates in the diet. Now, there is one more symptom called as malabsorption syndrome, where there is deficiency of some of the enzymes that are required for digestion of disaccharides. So, what will happen? Impaired digestion of disaccharides, malabsorption of disaccharides, that will result in the growth of the bacteria, inside in the large intestine. And as a result of that, there will be increase in bacterial byproducts and mucus production that will lead to the injury to the small intestinal cell wall. So the chain of events occur like that, and if this is not treated properly or immediate action is not being taken place, it will lead to serious situations. Next, metabolic fate of lactose intolerance. No, lactose intolerance. The lactose intolerance is a condition that is commonly seen in many situations. Usually in the kids, lactose intolerance is very commonly seen. This is usually because of deficiency of lactase. Sometimes what will happen, the lactase quantity is very less, the enzyme quantity will be very less, and when the children consume more and more of the milk, some amount of the milk won't be able to get digested because of deficiency of lactase. The lactase, when there is deficiency of lactase, the lactose will get accumulated in the gut. That will lead to irritant diarrhea and flatulence, because we have already seen what will happen when any carbohydrate is not digested. It will attract the bacteria. Bacteria will grow, hydrogen and all the gases are formed. It will lead to acetic acid, butyric acid and lactic acid. All these acids are produced. As a result of that, it will lead to irritant diarrhea and flatulence. Two types of reasons can be done. One is congenital, another one is acquired. Congenital, by birth they will have. Now acquired is usually as the age advances, people will have less. Like when you were in school, you used to consume milk. Now you are in college, you won't be able to drink milk. Sometimes if you drink milk also, not many but few of them, when they milk, they feel the bloating sensation and everything. That is because the activity of the lactase is decreased, because we are not being taking milk for such a long time, like milk just like that, without adding anything in that, milk just like that we are not taking from a long time. As a result of that, the lactase production might have been, it is not that decreased, but that will lead to little amount of lactose intolerance. That doesn't mean that it is a chronic disease. Abstaining from milk for a very long period of time and then you consume milk, that will lead to a little bit of lactose intolerance. What is the best way to treat this lactose intolerance is providing curd. Curd will have lactobacillus that will curb all these conditions. So this is a clinical significance that is associated with lactose intolerance. You have to understand why there is flatulence, diarrhea in lactose intolerance conditions. Now, we'll see what is the fate of, what will be the metabolic fate of any carbohydrate. Now, so finally, what we have seen in digestion absorption, whatever food we take, if it is even very complex polysaccharide, what is absorbed into the intestinal epithelial cell is only glucose, galactose and fructose. So glucose, galactose and fructose will enter into the portal circulation. Then once it enters into the cell, the glucose will get phosphorylated to form glucose 6-phosphate. The fructose and galactose also get phosphorylated. Then after that, after few reactions, they will get converted to glucose in the liver. Then the glucose 6-phosphate that is formed, it can have many goals to meet. It can undergo glycolysis and produce ATP and NADH, or if the glucose is very much excess, it can get stored as glycogen by the process called as glycogenesis, or it can go into HMP pathway to produce reducing equivalents and ribose, where it will be producing NADPH+H+. So now in the next class we will see we'll talk about the glycolysis.